Good morning, and thank you for joining us today. I'm Dr. Y.F. Niao from the Center for Research on Communicable Diseases in University Tunku Abdul Rahman. I have the privilege of moderating this webinar on COVID-19, the virus and the host. This current pandemic has generated so much fear, anxiety, and confusion that we need to have more discussions based on scientific and medical evidence to get a better understanding of the mechanics driving this pandemic. It is mortifying, I think, that despite all the technological advances we enjoy in our modern world, we can still allow a little virus to spread so far and wide and to cause so much of ill health and socio-economic upheaval worldwide. What is so special about this virus that makes it so fearful? Where did it come from? Is it man-made or natural? How is it evolving? How does it attack us? How does our body respond to its presence? And more importantly, how can we prevent infection or get over an infection and not succumb to it? In this webinar, we are fortunate to have two speakers to help address some of these issues. They are Assistant Professor Dr. Yap Mo Lan from the Faculty of Science in Utah and Associate Professor Dr. Pang Yong Kek from the University Malaya Medical Center. Dr. Yap is a genetics and molecular biology graduate from University Malaya. She obtained her PhD in bioengineering from the Tokyo Institute of Technology, and since then she has been studying the structure of very interesting viruses, including the sensational Zika virus associated with microcephaly in newborn infants, and dengue and chikungunya viruses that are more familiar to us in Malaysia. Dr. Pang Yong Kek is a senior consultant respiratory physician in the University Malaya Medical Center and the current president of the Malaysian Thoracic Society. Despite his heavy teaching and clinical duties, Dr. Pang has managed to participate in a large number of research projects and clinical trials. His main research interests are in obstructive airway disease, interstitial lung diseases, lung cancer, and of course lung infections, especially tuberculosis, but also viral infections in the respiratory tract. He has published a number of papers on coronaviruses and rhinoviruses that are the most frequent causes of the common cold. So now let's proceed to Dr. Yap's talk on the virus that is the cause of COVID-19.
Okay, thank you, Prof. Mia, for the kind introduction. So I'll be talking on the virus that caused COVID-19. So this virus has been named as the Severe Acute Respiratory Syndrome Coronavirus 2. In short, we call it SARS-CoV-2. All right, in general, coronavirus is RNA virus. So it has a positive sense, single-stranded RNA molecule in a linear form. So in terms of the size of the genome, it is rather big, about 30 kilobase per. So kilobases. So it is possibly among the largest in the RNA viruses. So this RNA molecule interact with the capsid protein to form a nucleocapsid. So it is arranged in a helical way. In terms of the shape of the virus, so you can see on the right, which is the electron micrograph of the virus particles. So the shape itself is rather irregular. So it is roughly spherical, but not exactly spherical. So we call it as pleomorphic virus. So the diameter itself, they have about 50 to 160 nanometer in terms of the diameter. And it is an envelope virus. So it has a lipid bilayer membrane surrounding the nucleocapsid. So if you look at the outer surface of the virus particle, you could see these crown-like projections. So this is the spike proteins, the spike projecting or presented on the virus surface, the surface of the virus. So it is a very important protein. So it is the main component for contacting with the host cell receptor. So it is important in a way that it will be serving as the component that interact with the host cell. So coronavirus circulate among mammals, bats, birds, and humans. So they cause respiratory infections. All right. In terms of the discovery of coronavirus, it is backdated in 1930s. So it was probably first discovered in infectious bronchitis chickens, and later in 1940s, they were found again in this hepatitis mouse. But only in 1965, then the human coronavirus was first isolated, and it was first observed under electron microscope by Dr. Jun Almeida, this lady here, and they published the paper in 1967, which is the year of the naming of coronavirus officially. So it is based on or referred to the unique features of the crown-like projections, the spikes on the surface of the virus. Okay, there are 7 types of human coronavirus been documented so far. So these first 4 have been circulating in human population for quite a while, and they cause only mild infections, and a lot of time they cause common cold as well. So it's very likely we get a coronavirus infection before, but we just do not aware of it because they just cause common cold that is known to be caused by rhinovirus as well. So only until 2003, then we have an outbreak that caused by SARS-CoV that give us the SARS disease. Then we noticed that human coronavirus do kill people. And 9 years later, we have another outbreak in Middle East that give us MERS disease. So this caused by another human coronavirus. We call it MERS-CoV. So rather recently, in the end of 2019, and we are still suffering from that, we got another outbreak that gave us pandemic around the world caused by SARS-CoV-2 that gave us the COVID-19. So these 3 human coronavirus do kill people. So in terms of the origins of the virus, we found that among 7, including the latest one that we have, most of them do come from bats as the natural host, and some of them do come from rodents. But they do need an intermediate host to transfer the virus from bat to human. So for example, in SARS, we have the civet cats to transfer the virus from bats to human, and in MERS, we do have camel as the intermediate host. But unfortunately, for this new virus that we have, SARS-CoV-2, we still in the search of the intermediate host, because we haven't actually been able to isolate the identical virus from a wild animal. So that for that, we still haven't been able to confirm what is the intermediate host that carry the virus from bats and transfer it to human. Okay, in terms of the naming of the virus and the disease that caused by this virus, we have 2 authorities or organizations that are responsible for the naming. So ICTV refers to the International Committee on Taxonomy of Viruses. So in this case, it has to go to the Coronal Virus Study Group, CSG, for the naming of the virus. So this new virus has been named as SARS-CoV-2 because it is highly related to the SARS-CoV that was first discovered in 2003. So they both put under the species of SARS-related coronavirus. And in terms of the disease that caused by this virus, WHO was the one to name the disease. So they named the disease caused by this virus as Coronavirus Disease 2019. And in short, we called it COVID-19. I think everybody knows it and everybody's talking about it. But to be more precise and accurate, especially when it comes to scientific research, we want to be clear that the name of the virus is SARS-CoV-2 and the disease itself is COVID-19. Okay, in terms of the fatality rate of these 3 human coronavirus that kill people, SARS do have around 9% of fatality rate, but it is not really that high if we compare it with MERS, which has 37% fatality rate. But for COVID-19, we still do not have an exact idea yet because it is still ongoing and we have so many cases that hasn't have a conclusion yet. But for the moment, we observe that it could be probably around 10%, but hopefully at the end of the pandemic, we do have lower fatality rate. But what actually surprised us is that if we look at the number of cases, COVID-19 have way too many compared to SARS and MERS. In fact, that indicates that the transmission rate of the virus is much higher compared to SARS-CoV and MERS-CoV. So this new virus probably have very different features on the virus itself to actually increase the transmission efficiency from human to human. In fact, for SARS and MERS, human-to-human transmission needs very close contact in order to transmit the virus. But of course, for COVID-19, we know that the virus can transmit very efficiently. Okay, this is just the official classification of SARS-CoV-2. So it is under family of Coronaviridae, and it has been classified into the species of SARS-related coronavirus, and it has been given the name as SARS-CoV-2. Okay, this is the first image taken by using electron microscope of the SARS-CoV-2 particles. So I think this is backdated in February 2020 this year. So China was trying to look for the agent or the pathogen that responsible for the disease, the new disease that occurred in Wuhan. So this is the first image that they took on the virus, and they could then confirm this disease is actually caused by a virus. So in couple with the genomic or genome sequence, the genomic information, so we are pretty sure that this pathogen is a virus and that it belongs to the SARS-related coronavirus. So we have to actually combine both evidence to confirm what is actually causing the disease. So if we look at the genomic organization of SARS-CoV-2, a few non-structural genes and 4 structural genes. So when we have this genomic information, so we do a blast search. That means that we try to compare the sequence to what we have in the database. And what we find out is that it has a very good match, about 80% in terms of the full-length genome, to SARS-CoV that was found in 2003. So from there, we know that it is a new virus, but not exactly the same, but closely related to SARS-CoV-2. What's more is that later we found out there is a bat origin SARS-related coronavirus has even higher, 96% sequence identity. Proof that, or it is a very strong evidence to show that is higher than 93%, and for the amino acid sequence, it has identity almost most of them higher than 98%. So if we draw a phylogenetic tree to show the genetic distance comparison among the coronaviruses, so for this case, we use the RNA-dependent RNA polymerase gene as a comparison. So this gene is very conserved, and it is a good candidate to draw the genetic distance among viruses. So it gives us an idea that it is very closely related to the bat's origin SARS-related CoV. And it has a slightly distance from SARS-CoV, but it is even more distance from MERS and other human coronaviruses. All right, if you look at the structure of the SARS-CoV-2, okay, this is just a model. It is not an actual particle. So we could see that on the surface of the virus, we do have this spike protruding out. This is what I showed you just now, is the crown-like projections. So in fact, it is a spike protein. Actually, 3 copies of the spike protein form one spike that protruded out from the surface of the virus. All right, this is a very important structure that will get contact with the host cell's receptor. And membrane protein, it is quite abundant and on the surface of the virus as well. And then we have less copy of the envelope protein. So if we look at the cross-section of the virus particle, so if we make a cut and then look into the internal view of the virus, we can see that this is the nucleocapsid that was arranged in a helical symmetry and nicely packed in the internal side. This is probably the one that contact with the nucleocapsid and then embedded in the membrane, and this spike protein protruded out from the lipid bilayer membrane. All right. So I do like to briefly show you the life cycle of coronavirus. There are 2 points that are particularly important: the first step and the second step, which is the entry, attachment, and the fusion step of the virus. So these 2 steps involve spike of the virus, which is responsible to recognize the receptor on the host cell. So in this case, we take ACE2, which is the angiotensin converting enzyme 2, a molecule on the human cell surface, and it is the receptor for this SARS-CoV-2 virus. So the spike protein, the spike on the virus, has a particular domain, what we call the receptor binding domain, that has the sequence to recognize the molecule, ACE2 molecule, and it will bind and attach to the ACE2 molecule. The next thing would happen is that endocytosis of the whole particle into the host cell. So there will be a vesicle form surrounding the virus particle, but the virus particle are still intact. So when the vesicle go along into the cell, the pH will get lower to acidic pH. So this will trigger the next step, which is diffusion of the virus to the host cell. So what happened is that the low pH will trigger ferritin, which is a protease, that will make a cut on the spike protein. So there is a specific site on the spike protein that will be recognized by the ferritin, and it will be cleaved. So this cleavage will then expose another part, which is a fusion peptide on the spike protein that meant to fuse with the host cell membrane, that will lead to an opening of the particle and release the RNA molecule into the host cell. So from there onwards, all the replication will happen to make more RNA molecule and to make more proteins that give us new viruses. So for these 2 steps, the spike protein evolved. So when we come to talk about vaccine design, so what we would like to do is that we probably want to see the life cycle, the first step of the virus will be blocked. So when we have this whole virus to come into our body, our immune system probably will generate antibodies against the most exposed protein of the virus, which is here in this case is a spike protein. So if we would be able to get antibodies that bind to this protein, the spike, that will then stop the virus from attached to the ACE2 receptor molecule. So if this step work, then of course the vaccine actually could protect us. If it doesn't work, then we hope that the next step, the fusion step, will be blocked. So that's where we target the second region, which is the fusion peptide on the virus, will be recognized by antibody as well, so that the fusion wouldn't happen, and then the RNA molecule would not have been released into the host cell. Then the replication will be blocked as well. So we will go into this spike protein a little more detail. So showing here, I'm giving you on single polypeptide of the S protein, the spike protein. So if you remember, I mentioned just now we need 3 copies, 3 polypeptides of the spike protein in order to make 1 spike. But showing here is just 1 copy, just to make things clearer. So it is a fusion protein, class 1 fusion protein. And some features is it has helixes, and then how it positioned is that it is perpendicular to the surface of the virus. So what is important here is that it has a receptor binding domain, color in cyan here. So this is the particular site that will be binding to the ACE2 receptor. So what is more specific is that at the tip of the receptor binding domain, we have this receptor binding motif, which has the interface that interacting with the ACE2 molecule, the receptor. So this spike protein has a pre-fusion state, and we have 2 conformations, closed conformations and the open conformation. So the difference is that the closed conformation will have this receptor binding domain not exposed, but in open conformation, this receptor binding domain will be exposed, exposed as well, and it will be able to bind to the receptor. So according to the group that worked out this structure, the interesting is that in the closed conformation, all 3 copies of the spike protein have the receptor binding domain put down, not exposed. However, in an open conformation, only 1 copy of the receptor binding domain is standing upright and exposing the receptor binding motif. So which means that probably only 1 receptor that binds to this spike protein would actually be sufficient to trigger the next step of the infection. So I'll be possibly, So this is the receptor binding surface. Showing here is the structure of the receptor binding domain, and then on top and at the tip part, it is the receptor binding interface. So shown in red is the sequence for SARS-CoV-2, and shown in blue is the sequence for SARS-CoV. So if you compare the structure, SARS-CoV and SARS-CoV-2 have very similar, almost exactly the same, but very similar. We would say highly similar in terms of the structure of this receptor binding domain. And if we look at the sequence, so there are 17 or 16 contacted residues from these 2 viruses that highly contact with the ACE2 receptor. So they both have share similar residues for contacting with the ACE2 receptor, which means these 2 viruses, they recognize ACE2 as the receptor. So if we could find a vaccine, or we could make a vaccine against this receptor binding domain, that will probably work for both viruses. So which means that it gives us protection against 2 viruses at the same time, if we will be able to have a vaccine against this particular region. Okay, so this is electron density map actually, but we colored only the receptor binding domain that is in the open conformation, which is standing up, and this is the receptor binding motif, which is exposing, whereas the receptor will bind. So color in this, those color in red, this orange and green, will be the potential binding site for antibodies. And then here it is color in blue, which is the position of the fusion peptide. So that will be possibly bind by the antibodies. So if we'll be able to have antibodies that target on this region, so it's very likely we could block the attachment or the binding of this virus to the receptor. So when we design a vaccine candidate, we probably want to focus on this spike protein, and then particularly on the receptor binding domain. And of course, another option will be the peptide. So I have a few movies to show you how the spike will interact with the ACE2 receptor. So we will see the first one. So this is a spike, okay, consists of 3 spike proteins. And green and the blue ones are the protein in the closed conformation, and then the one in orange, it is in the open conformation. It has this binding receptor binding motif that is exposing. So when we have this receptor to come in, hold on. Okay, this is the receptor, ACE2 receptor to come in. So it will recognize this receptor binding domain, and then the motif here would serve as the interface to actually interact with this ACE2 receptor. So this is how it would look like. And I will show you in both open and closed conformation with ACE2 receptor. So this is an electron density map of the spike in the closed conformation here on the left, and in the open conformation on the right. So different color means different polypeptides. But in the open conformation, we have this receptor binding domain standing upright. So if we superimpose, showing in closed conformation and open conformation. So only one receptor binding domain will stand upright. Okay, so here comes the ACE2 molecule, and then it is how it would bind. This is how it would bind to the spike. So one binding would be sufficient to trigger the next step of the infection. Okay, so if we look at the next one, I do like to see how the full length of the ACE2 receptor on the membrane would probably bind to the spike on the virus. So this would be how it probably would look like. So spike in the open conformation, and then this is on the right is the ACE2 receptor full length. Presumably they are both on the membrane, and this is how it would look like. So the ACE2 receptor is a dimer, and then this would be how they would interact. Okay, so this is the binding site where these two molecules will interacting with each other. So in fact, one binding would be sufficient. Okay, I think this is pretty great movie to me. Okay, for vaccine development, we could see that there are a lot of different platforms. So some use the whole virus, either inactivated or weakened. Some do use a viral vector or nucleic acid. And some do use only the protein subunit, or they even make virus-like particles. But up to May 2020, we have more than 100 vaccine candidates that are under development. So out of that, 12 are in human trials, and only 5 are now currently in the phase 2 clinical trial. But this is considered as pretty fast development in terms of vaccine development. But what I want to emphasize here is that you may actually have a closer look, and then we find out all these candidates in phase 2 clinical trials, they are targeting on the spike protein. Either they are using full length or inactivated virus, which also have the spike protein, or for the one that from Germany, they use only the receptor binding domain, which means like they recombinantly produce the receptor binding domain and then inject it as a vaccine candidate. So that will trigger the immune system to generate antibodies that specially target on this particular domain. So I think lastly, I do like to show you a movie, how the antibody will bind to the spike, and then it will block the binding of ACE2 receptor. So in this movie, I will have 2 Fab, which is part of the antibody molecule, to bind to the spike. So again, on the left is the spike, and then on the right is the spike bound to two antibody. So this is only the fragment. So you will see this cyan and the yellow part are actually the antibodies. So two different monoclonal antibodies, and they actually bound just to show the binding, how it would work on the spike. So when it is in the open conformation, again, the antibody will target on this region or on this region. Okay, so the binding of the antibody will actually cover the binding site for the receptor. All right, this is how, if the receptor will come in, it will bind this way. But if you see, there are clashes in between the ACE2 and the antibody molecule. So which means that if the antibody will come in first and bind to the spike, the receptor will have no chance to bind to the spike. So that will prevent the attachment of the virus to the ACE2 receptor. So this actually also give you an idea how the monoclonal antibody treatment will work. So China is now producing the monoclonal antibody as a treatment given to the patient. So this will be a picture that probably this treatment would work. So if you have a monoclonal antibody that actually subjected or against bind to the this particular spike region, that will actually block the infection to happen. So it will help for the COVID-19 patient to recover. So I think this is pretty much I would like to share with you in this talk. Thank you for your attention. Yeah.
Okay, good morning, everyone. It's nice to meet everyone virtually, although I think I would like to actually meet all of you face to face, but I think we have to contend with the new normal from this point onwards. So it will be actually for quite some times, I think, until everything is back to the previous normal. Whether it will be back to previous normal, I'm not too sure, okay? But I think we have to live with the normal. Thank you very much, Prof. Niel, for the introduction as well as for the invitation to share the clinical manifestations of COVID-19. I think we have heard a lot just now regarding the virus itself. Now we would like to actually talk about the host, which is actually the clinical manifestations. This is the content of my talk. We'll give some introduction and move on to talk about the symptoms and signs, the imaging and laboratory findings, organ-specific and systemic manifestation, and also share with you some of the unique features that you only see in COVID-19 and much less in the other infection. And lastly, how do we capitalize on this understanding? Okay, first of all, COVID-19, the introduction. As you know that after the peak in April, now we are seeing gradual decline of the new cases. At certain days, we are seeing single digits, certain days we are seeing double digits. But nonetheless, I think it is on the downward trend, and I hope that this trend will continue, and hopefully we can suppress the virus completely one day. So you may wonder that why we still have this kind of a talk as the infection is going to be controlled. Well, we may think that the virus will be controlled in Malaysia, the infection. But worldwide, we are seeing that many other places, the virus is still ravaging across the world. So unless the whole world, the virus is under control, I don't think we can actually go back to our normal life. So you have actually learned a lot this morning regarding the culprit virus, which is actually the coronavirus, known as a SARS-CoV-2. So some of you might know this very well, even before this, but to certain people, they wonder if they have not heard too much about the coronavirus. So many people have heard a lot on like flu, the influenza, not so much about coronavirus. But we know that this is not something new. I think just now Dr. Yap have shared with you that this is not something new in the world. I think in the end of 2002 and 2003, we have witnessed how SARS, the first wave of the coronavirus, which have wreaked havoc in the world, particularly in Asia Pacific region. So the question then was, can it be stopped? Okay, the new age of epidemics. So we thought the virus was under control after about a year, but then now we are seeing actually a new wave of the surging of coronavirus. So before this coronavirus 2, the SARS coronavirus 2, we are also seeing another coronavirus that is ravaging the world through the transmission from camel to humans, and then from human to humans. So the first case was reported in 2012, which is about 10 years after the SARS. And then this was believed to actually start from the Middle East. And you can see here from the map that it has also spread to many other parts of the world. So it doesn't look like we are actually getting the coronavirus under control. Every now and then, new coronavirus is emerging, and then it is causing widespread outbreak in the world. And the trend is that it is coming up, seems to be coming up every 10 years. So the first one was 2002, and then 2012. And then now we are seeing this in 2019. So it's actually less than 10 years. While coronavirus is not the only infection we need to be concerned, there are many other microorganisms, such as influenza, which is causing seasonal outbreak every year during the winter. And we also have the RSV, Ebola, and bacteria such as tuberculosis. So I think the battle with all these microorganisms is not going to end. So certainly, even if we can actually control this COVID-19, but I think it's not the end of the story. I think it actually makes us wonder actually, how are we getting ourselves better prepared for another future outbreak. And I think that the lessons learned from the past, as well as the present pandemics, will certainly enhance our understanding of this long-term enemy. And hopefully, with the better understanding gained from these battlegrounds, it will form actually the foundation as well as guidance in making us more prepared for any future outbreaks, not just limited to the coronavirus. Now I would like to move on to the clinical manifestations of this COVID-19. We talk about the symptom first. As you know that the common symptoms manifested by COVID-19 is fever. This is actually the data from China, mainly from Wuhan. They found that fever was one of the commonest symptoms on the admission. Up to about 43.8% of them had fever on admission. For those who did not have fever, some of them actually developed fever after hospitalization. So you take that into consideration, admission as well as hospitalization. It is very common, up to 88.7%. Cough is the second commonest symptoms. This is not surprise because the site of the entry of the virus into the body is into the lung or into the airways. So obviously, when the lung is inflamed, cough is going to appear in the affected individual. Fatigue, which is like the tiredness, I think is quite common to any viral infection. Sputum production next, as well as shortness of breath. In that cohort, about 18.7%. And then less common symptoms like myalgia, arthralgia, sore throat, headaches, and chills. So if you look at this list, it's very much similar to any other viral infection, including flu or influenza. So it's actually very difficult clinically to distinguish the COVID-19 from other viral infection. It was also reported that apart from the respiratory and systemic symptoms, patients have other symptoms such as nausea, vomiting, nasal congestion, diarrhea, and some may have abdominal pain, as well as conjunctival congestion. So apart from actually what was reported in China, I think European cohorts also highlighted some unusual symptoms that are not traditionally associated with viral infection, including the SARS in 2003, which is the anosmia or hyposmia. Anosmia means a loss of smell. Hyposmia means a reduced the smell sensation. This juice here is actually alteration in the taste, and some was also noted to have confusion. Then let us look at what are the clinical manifestations in terms of the signs. Signs, I think symptom is actually what complained by the patient. Sign is what the doctors identified. And of course, I think fever is a symptoms as well as a sign. And essentially, the sign is not very helpful. I think the symptoms are probably more useful in identifying the infection because the sign, apart from fever, the rest are virtually quite uncommon, right? From judging from ranging from throat congestion, which was only about 1.7%, tonsil swelling, and large lymph nodes, skin rash. You can see that the percentage is, apart from fever, they were less than 5%. And of course, I think the other important signs is to look for sign of pneumonia. I think such as like a bronchial breath sound, crepitation, or auscultation. But the pneumonia in COVID-19 is actually quite different from other bacterial infection, whereby the predominant manifestation is the ground glass changes. And the ground glass changes typically do not give rise to many signs, like what I mentioned just now. The bronchial bleeding as well as crepitation is typically absent from the chest. So that actually add another layer of difficulties in identifying the disease early. So we have to resort to other imaging tests, such as chest X-ray, as well as CT scan. So you can see here, chest X-ray, I think is quite useful. Especially in the Wuhan cohort, you can see that any abnormality seen is up to about 59.1%. And if we resort to CT scan, it is much more sensitive. 86.2% of the patients that the doctors see over there had some CT scan changes. Just now, I mentioned that the ground glass opacity is one of the commonest findings in imaging. But please take note that chest X-ray is not very good in identifying ground glass opacity. So only 20.1% of them was visible on chest X-ray. Whereas CT scan is a much more sensitive imaging modality. So you can see here 56.4% of them can be visualized on CT scan. Apart from ground glass opacity, there were other changes such as local patchy shadowing, 28.1% in X-ray and 41.9% in CT scan, and bilateral patchy shadowing. Okay, so around 30 to 51%. So I'd like to show you two chest X-ray for the same patient. You can see on your left, that is actually a patient with COVID-19 on day 1 of admission. If you are familiar with chest X-ray, okay, there are some actually changes seen on the chest X-ray on day 1 of admission, consisting of ground glass changes as well as patches of lung consolidation. But after day 9, you can see that some of these ground glass changes disappearing. What you are seeing more here is a patchy opacity, and some of them appear a lot more rounded because the inflammation is getting more organized by then. Okay, and then CT scan, much more sensitive imaging modality. On the left, what you can see patches of ground glass as well as consolidation. And on the right, you can see the changes are a lot more extensive, consisting also ground glass changes, consolidation, and please take note that pleural effusion is not a common manifestation of COVID-19. Only about, I think, 3% of the patient with COVID-19 had pleural effusion. What about the laboratory results? Like any viral infection, the white cell count could be either high or normal or low. So what we can see here is that the typical changes are the low white cell counts compared to a raised white cell count. And of course, vast majority of them actually had a normal white cell count. For patients with raised white cell count, you can see that only about 6% of them had raised white cell count, and about a third of them had low white cell count, less than 4,000 per mil. Okay, and then for patients with a more severe disease, the white cell count is typically much lower. And on top of the white cell count, the lymphocyte count also is very useful. You can see here that up to about 83.2% of them had low lymphocyte count. And then, of course, if the disease is more severe, the drop in the lymphocyte count is much more severe. And platelet counts, about a third of them had a drop in a platelet. The more severe they are, the more likely the platelet count is below the normal. So more than half of them actually had low platelets. So it's just like other viral infection, including influenza or dengue and all this viral infection, the platelet count is low. And in terms of other laboratory tests, useful tests to gauge the inflammatory markers, such as C-reactive protein, was shown to be elevated in about 2/3 of the patient. And the more severe the disease, the C-reactive protein is going to raise higher. So I think this is a very useful marker to gauge the level of inflammation in the affected individuals. and also to gauge the severity of the disease. Procalcitonin is useful to identify the bacterial infection, but for viral infection, it is not very useful. You can see that only 5.5% of them had raised procalcitonin, so it is not very useful. Lactate dehydrogenase, it is raised in more than a third of the patient. And for patients with more severe illness, obviously we are going to expect the lactate dehydrogenase to raise higher. Liver enzyme such as aspartate aminotransferase was also raised in about 1/5 of the patients. Interestingly, the D-dimer is also raised in about 40 to 50% of the patients. And the more severe the disease is, the more likely the D-dimer is going to raise higher than normal. I think we have learned a lot this morning regarding the mode of entry into the cells. So the virus gain the entrance into the cell via the attachment of the spikes with the ACE2 receptor. Just would like to highlight that beside the lungs, other organs such as heart, intestine, kidney, as well as the brain also contain ACE2 receptors. So there's no wonder that beside the lung, the other organs are also affected, because the virus can also gain entry into the other organs through the ACE2 receptor in the other organs. And like any other viruses, this organism do not have machinery to replicate itself. So essentially, after the entrance into the cells, it will use the machinery in the human cells to make new viruses. This is also, I think, the mechanism that Dr. Yap mentioned just now, and this provides the opportunity for us to block the replication of the viruses. So in order to fight the virus, we know that our body has the innate immunity to control as well as to eliminate the virus. So we do not need like a specific drug to treat the virus. It's just that this innate immunity takes sometimes to develop. So by then, the virus may cause a lot of damage and havoc in our body. So I think this is important to understand. And the question is why some patients had more severe disease than the rest, and why certain patients were quite asymptomatic compared to the rest. We believe that it is due to some dysfunction in the immunity. Dysfunction means that either the immunity is not strong enough, or the immunity is overreacting. So we know that many of the damages seen in the cells as well as the organs are in fact caused by the inflammatory cells as well as the chemicals released from our body, okay, and known as the cytokines. So we know that before a full-fledged immunity developed, during the transition period, the function as well as the tissue will continue to suffer the damages. So this is a graph showing the clinical cause of COVID-19. So what you can see here that this author suggests that maybe the stage of the infection can be divided into the early infection, which is the stage 1, stage 2, the so-called intermediate stage, or known as the pulmonary phase, and stage 3 is a hyperinflammation phase. Of course, this is an oversimplified graph. Not every patient go through the similar phases in a similar extent, but it does give us a glimpse of actually what's happening in our body. So what you can see that after infection, the number of the virus is going to increase exponentially. So when the virus is increasing, the host is also mounting the immunity against this virus. So eventually, the virus is under control because of the immunity that is developed to counter the virus. If the inflammatory response as well as the immune response of the host is kept unchecked, the inflammation can indeed cause continuous damage to our human body as well as the organs function. The natural history of COVID-19 in a way is quite predictable. Okay, so I've shown to you that during the early phases, replications occur in the body, particularly in the upper airway. After that, it moves on to the lower airway, where it is causing lower respiratory tract infection or pneumonia. And then subsequently, it will spread to the other part of the bodies. These viruses typically have a very short incubation period between 3 to 5 days. Some mention like it ranges from 2 to 14 days. The natural history is quite predictable. The onset of symptom to dyspnea usually take about 6 days, and onset of symptoms to hospitalization is about 8 days, and then to intensive care is about 10 days. Now let's move on to talk about some unique clinical manifestations of this disease. So in a study performed by Julian and al., among the 107 consecutively confirmed COVID-19 patients admitted into ICU for pneumonia. So we are talking about patients who have more severe disease, who require ICU care. Interestingly, to see that there was a high percentage of patients who had pulmonary embolism, so 20.6%. Essentially, 1 in 5 patients had pulmonary embolism. And this was compared with equally severe non-COVID patients in ICU. And the frequency of pulmonary embolism in this control was said to be only 6.1%. So what you can see here, this represents an absolute increased risk of 14.4%, which is about 2 times more common than other critically ill patients in ICU who do not have COVID-19. And then what about the histological changes? So this is a study performed by Ackerman and al., where they perform autopsy, especially on the lung tissue for patients who pass away because of COVID-19. So 7 of them were obtained. This is to compare with another 7 samples for those who die because of ARDS secondary to influenza A infection, as well as another 10 age-matched patients without the lung infection. I mean, they died secondary to some other causes. So what are the findings from this autopsy of the 7 patients? They found that all patients had diffuse alveolar damage with necrosis of the alveolar lining cells, the pneumocyte type 2 hyperplasia, as well as the linear intra-alveolar fibrin deposition that you see on the right of the slides. Okay, this is actually what it looks like in the autopsy. In 4 cases, the changes were focal with only mild interstitial edema. The remaining 3 cases had more global involvement. And then they look at actually the ACE2 expression in the epithelial cell. They found that for COVID-19 as well as influenza, there were high number of ACE2 expression in the epithelial cells compared to the control. It is not only the epithelial cells, which is the alveolar cell that is affected, the endothelial cells, which is the inner lining of the blood vessel, also showed high expression of the ACE2 receptor. And in addition, they also found that the lymphocytes, the extracellular lymphocyte also had high ACE2 expression. And just now I have highlighted to you that patients with COVID-19, they have a very high risk of blood clot forming in the blood vessel. And then this blood vessel, if it's in the vein, it may migrate and block the vessel in the lung, causing pulmonary embolism. I've also mentioned the study just now. Those who were critically ill require ICU admission. About 1/5 of them had pulmonary embolism. So for the study on autopsy, the expert also found quite similar findings, but this time they found thrombi in the pulmonary artery of 4 of the 7 patients with COVID-19 as well as influenza. In addition, they also found that these microthrombi were 9 times as prevalent in patients with COVID-19 compared with patients with influenza, suggesting that while any infection, you have a higher risk of the blood clotting in the blood vessel, but for COVID-19, the risk is far much higher, right? And not only the artery, they also found a blood clot in the capillaries as well as a vein of 3 COVID-19 patients. Apart from the thrombi seen in the blood vessel, they actually used electron microscopy as well as the microvascular corrosion casting to look at actually the vascularity of the blood vessel in the lungs, and they found that by and large the capillaries are deformed and also elongated with a changing a finding called the intersusceptive pillars between the capillary. And this is the process of a new vessel formation, suggesting that not only the capillaries are deformed, but there are more new vessel formation in the lungs of the affected individuals. And you can see that the graph over here, they compare COVID-19 versus influenza versus control. Top on the list, of course, is COVID-19, followed by influenza, and the control virtually have very little new vessel formation. And the other thing we'd like to share with you is regarding the pediatric inflammatory multisystem syndrome associated with SARS-CoV-2, or known as PIMS-TS. And we know that during this pandemic, there were some children who developed fever and multi-system inflammation. I think this finding were noted particularly in the European as well as the Americans cohort. I think we did not actually hear much from the Chinese cohorts. what we heard most is from the European and Americans cohort. And some of these children were critically ill with a shock, as well as a multi-organ failure, and some of them required intensive care. Interestingly, this manifestation has been linked to the Kawasaki disease in children, or the Kawasaki disease shock syndrome. And there is a cohort published on this syndrome by Whitaker and al. 58 children with this syndrome were analyzed, and they found that all of them had fever and non-specific symptoms such as abdominal pain, skin rash, conjunctival injection, and 50% of them developed shock and required inotropic support, as well as fluid resuscitation. 22% of them met diagnostic criteria for Kawasaki disease, and 14% had coronary artery dilatation or aneurysms. And these are the case definition for the PIMS TS, or the multi-system inflammatory syndromes. I think for those of you who are interested, can actually search for it. We have WHO definition, we have the British definition, we also have the CDC definition of this syndrome. So what I would like to highlight is that the key features used for diagnosis is that the patient age must be less than 21 years old. They should have fever for at least 3 days. There must be evidence of inflammation involving more than 2 organs. There must be no evidence of other microbial infection apart from COVID-19. Some actually required the inflammatory markers to be raised. This syndrome can be diagnosed with or without the presence of the confirmative test of COVID-19. In terms of differences between this multi-system inflammatory syndrome and Kawasaki, as well as toxic shock syndrome, we can see that the age group for this COVID-related multi-system inflammatory syndrome, the age group, the children generally are older compared to the Kawasaki disease. The lymphocyte counts was also lower in general. The D-dimer test is higher than the other two syndrome, and the C-reactive protein is typically higher as well compared to those with Kawasaki disease. So organs involvement, I've mentioned just now that apart from the lungs, ACE2 receptor is also present in many other organs. So it is not surprising that other organs such as the heart, the kidneys, the brain, the intestine, the liver are also commonly affected by the virus. So in terms of complication, luckily there are not many patients with septic shock seen among the COVID-19 patients. So by and large, the commonest complication is still the acute respiratory distress syndrome, or known as the ARDS. You can see here that for patients with severe COVID-19, 15.6% of them had ARDS. Okay, a few of them had acute kidney injury, and those who had severe ARDS, some of them may require invasive mechanical ventilation. All right, so we move on to the final part of the talk. We know that although there's no specific treatment for the virus, we can count on the body healing capability to overcome the disease. In some patients, supportive as well as prophylactic treatment, I think is crucial to ensure a better outcome for the patients. Okay, so what are the implications for COVID-19 treatment based on the understanding of the clinical manifestation? First and foremost, we know that for those who have a severe disease, vast majority of them had severe lung involvement, severe pneumonia or ARDS. And we know that once the lung become more congested with the fluid, the gas exchange is going to be severely compromised. So what we can do in clinical practice is that we can administer enriched oxygen for the patient. If this is not good enough, we may have to resort to the mechanical ventilatory support to ensure that these vital functions continue to sustain life. For those with very severe disease, whereby even with mechanical ventilation, the oxygen exchange is still not good enough to sustain life, some centers resort to using the ECMO or the extracorporeal membrane exchange to sustain the patient's life. And we know that hyperinflammatory response sometimes is also causing the problem or causing the damage or disruption to the function. So one of the commonly secreted cytokines is interleukin-6. Treatment with interleukin-6 has been shown in some non-randomized study to improve the outcome of the affected patients. Just would like to show you one of the observational control study whereby 78 received tocilizumab, which is anti-interleukin-6, and compared to 76 patients who did not receive this therapy. The group that received this therapy was associated with for the has inflammatory effects on the inflammatory cells. So what you can see here that there was a study done whereby a small number of patients with ERDS were randomized to receive the metaprednisolone versus those without metaprednisolone. And what you can see here in the survival curve, those who receive metaprednisolone seem to actually had a better survival, suggesting that perhaps corticosteroids do have a role in patients with severe disease. And this is actually quite different from the SARS in 2003, whereby systemic steroid has not been shown to be beneficial in patients with severe pneumonia. So this is actually something that is different of this virus compared to the SARS in 2003. And of course, I think we should not forget about the importance of certain supportive care, such as the fluid as well as the electrolytes. Why it is so important is that I think anyone with viral infection, generally their appetite is reduced. They tend to perspire because of the fever. The blood pressure may drop. Perfusion to the tissue may be compromised. So it is very important to ensure that their fluids as well as the electrolytes are maintained to ensure that the body, the organ is functioning properly with good tissue perfusions and electrolytes balance. And of course, in patients with renal failure, hemodialysis will temporarily replace this function to ensure homeostasis in the body. Prophylactic therapy, just now I've highlighted that this disease somehow had a very high risk of thromboembolism as a result of the damage caused to the inner lining of the blood vessel, or what we call the endothelial damage. And this result in the blood clot formation at the artery as well as the vein. And we know that if the clots occur at the arterial sites, this will result in stroke, heart attack, heart failure, as well as gangrene of the limbs. And the thromboembolism at the lungs may also be partially accounted for the respiratory distress or the ERDS. And then if they occur at the venous site, it causes deep vein thrombosis, typically occurring in the lower limbs. From there, we know that the clots may migrate to the lung vessel, causing pulmonary embolism. And hence, the treatment with anticoagulation is believed to reduce the risk of these complications. So with that, I'd like to thank you for your attention.
Okay, so we have questions for Dr. Yap first. The first three are, the first one is there any evidence of the virus evolving fast enough to affect effective vaccine development?
Hello, yeah, okay. So far, we do not have an evidence for that. Although we know that RNA viruses, they mutate or they evolve pretty fast. But in terms of vaccine development, so we have to pay attention to where the mutation will actually occur. So if, for example, go back to the talk I gave just now, the information. So if the mutation actually fall onto the binding site to the receptor or somewhere that actually critical, then it will probably affect the vaccine development. That means like if we do have a good vaccine, but yet after a while, the virus have a mutation on this particular critical site, then it probably will make the vaccine not work anymore. So it has to depend on the site of the mutation. So so far, but we have not come out for vaccine yet, and there's no evidence that this mutation will fall onto this receptor binding domain very frequently. Yeah.
Okay, so before we go on to the next question, I think we have a raised hand. Dr. Jeffrey Niao, would you like to pose your question?
Yes, my question to Prof. Yap is, there's been some talk about different strains of the virus, A, B, and C type, according to regional distribution. Apparently, the Asian type is just so different from the North American type that the Asian patient tends to have less severe symptoms. Can you explain or explore some reasons from the structural point of view?
All right. Thank you for the question. Okay. On this question, there is a paper trying to clarify it. They suggest that there are three types, A, B, and C. However, when the paper was published, and then there were some comments. Although I'm not the expert in studying this, but when we look at the comments, that it has been pointed out that we have to interpret the data more carefully, because when they try to categorize type A, B, and C, it was just based on a single difference, nucleotide difference. So we have to then look at if this single nucleotide difference do make meaningful changes to the amino acid sequence, which give us the protein that probably will function, play a certain role in certain function. So that is the critical part. But however, categorizing different type of the SARS-CoV-2 based on a single nucleotide difference might be a little risky. So there are comments saying that we probably have to take a look of this data more carefully. So I probably don't want to put too much comments on that, because I think that I agree with more careful examination of the data. So in terms of the statistic of how much the mutation actually relate to different symptoms, I think we probably have to also screen through more samples in order to make a conclusion out of it. Because for vaccine development, it is not really critical if it fall into somewhere else that not in the target or in the vaccine candidate. So it has nothing to do with the vaccine candidate. So that would be my answer. I hope I give you some clue.
Yes, thank you. But this question has a lot of political implications in terms of identifying R naught and trying to put the blame on where the source of the first case comes from.
Yeah, I agree, but it is more on the political side. I think most scientists or researchers will be more careful when it comes to interpretation of the data. So we will actually look at the scientific data and then do an interpretation and justification. I think most scientists or researchers do not agree with that. And there will be a question, say like if the virus is a man-made virus or it is a natural virus. I think most scientists will agree that it is a natural virus, because if we look at the features of the virus, it doesn't look like it is from man-made or from a lab. For example, if we do a prediction just solely based on the sequence, the computer will tell you the binding of this sequence or this virus to the ACE2 receptor is not good. So if one would want to engineer a virus, they will not take this sequence. But in fact, when we come to the experimental data, it showed that the binding of this SARS-CoV-2 bind much better than SARS-CoV. So this is something that we have to be careful with. So the prediction might not work so well or slightly unreliable, because it has the constraint of depends on limit to what we put it as a criteria to select. So when it comes to natural selection, I think the nature do a better job than the human being when it comes to selection. So there is a reason behind why this virus works much better than other coronavirus in terms of transmission. As I said just now, there are some features, but we have to look for some scientific data to support or to prove the fact that it is good or better than other viruses. Yeah.
Thank you very much.
Thank you.
Yeah, thank you, Dr. Yap. I think there's so much of political exploitation already. It's really depressing to read the social media. So yes, thank you for saying we must come back to science and clinical specialists for answers. Yeah. So this is a related question. We'll jump to question number 3. If you still see the questions on your screen, is there a HIV sequence in the SARS-CoV-2 genome? Because somebody take this, some scientist somewhere have written a report to say that because of the existence of this HIV sequence, this is a very good indication that the SARS-CoV-2 is man-made. What do you say to this?
Okay, yeah, I heard about this, but I think, again, I want to emphasize we have to be careful when we come to interpretation of the data. So for example, not to say for example, but HIV sequence do have certain similarity when it compared to SARS-CoV-2. This is because they share certain, say like genes. For example, the RNA-dependent RNA polymerase, the enzyme that make new molecule or RNA molecule from the RNA. So this protein do have a conserved structure. So when it comes to structure that do the same function, so they do probably code from the same amino acid sequence. So when it comes to this point, we will go directly to the nucleotide sequence, all right? So it might not be exactly the same, but the sequence of the nucleotide sequence probably will give the code, the codon that code for the same amino acids. So in a way that we probably will find high similarity on this particular gene, because it does have to give the protein the conserved structure and do the function. But of course, there are some differences in terms of the residues to suit or to make the protein fit better for this virus. So then that we find the differences in terms of the sequence. So if you want to solely base on the sequence, I think this has to be considered this point. And it depends on how much in terms of the nucleotide sequence that is included. So you don't go for like 10 nucleotides and claim that there's 100% identity, because 10 of them do match 100%. But 10 out of 30 kilobase pair, it probably doesn't mean so much. So we want to be careful when we do the sequence matching to see how much similarity or identity comparing in different viruses. But what I want to point out is that some viruses, especially like HIV and SARS-CoV-2, they are both positive sense single-stranded RNA molecule virus. So they probably do make use of certain proteins that do the similar function. So in that case, they do probably have some similar sequence. Yeah, it is not surprising to see that, but we would not claim this as man-made or engineered, because in natural, we do see this trend very common.
Yeah. Thank you, Dr. Yap.
All right.
So the next question is there evidence of cross-protection between SARS and the COVID-19, since they're so similar in the genome?
Oh yeah, it is possible, but if we want to talk about evidence, of course, we probably don't have it yet, or maybe I have not come across this. But as I showed just now, in terms of the structure of this particular region of SARS-CoV and SARS-CoV-2, they do have high similarity in terms of the structure. That probably show that the same antibody will probably recognize the same structure on SARS-CoV and SARS-CoV-2. So it is quite likely they will give cross-protection. If we have a vaccine, then it probably will protect us from SARS-CoV and SARS-CoV-2.
Yes, I think there's also some epidemiological evidence coming, maybe not evidence, but observations coming from previous countries which were previously affected by SARS. They seem to be less affected by COVID-19. That's according to reports. Right.
Those are more statistic. I think
yes,
we need to find the proof. Yeah.
Yes. So the last question for you is: COVID-19 seasonal? Will it disappear like SARS?
I think it is less likely to be seasonal. While we see that it happened in all over the world, in cold weather, or in Malaysia, we have sort of warm weather. We all have this, but I don't think it will be much affected by the weather. Yeah, but in terms of transmission, the condition itself probably limits the transmission. But if you want to say, would it be like totally go away? It is something that we have to observe, but it is less likely, I would say. Yeah.
Maybe it will appear like influenza coming once in a year or two. Okay. So, Iris, do we have further questions from the chat box? Anyone wants to raise hands again? If not, we go on to the questions for the second speaker.
Yeah, I have a few questions from the chat box.
Okay.
Now I'm sharing the screen. I compile it and share in the screen. Yeah.
I think this one is answered already.
See?
Yeah.
Then for the second slide,
shall we leave this to Dr. Pang?
Yeah,
Or,
sure.
yeah, would you like to answer this? Okay, maybe in the next one. Will the COVID-19 vaccine ever come to us? Is it as difficult developing it as for the dengue virus? Dr. Yap?
Oh, all right. Well, in fact, vaccine development most of the time take years. But for this particular disease, I think it is too serious, and then governments has sped up the process. So some even skip the animal test and then go to human trials directly. But does it really ever come to us? I think it's not too difficult to get a vaccine candidate, but the question is, does it work? This is the most important thing. It's not too difficult to get a vaccine candidate, but the main question is, does it work, and how well does it work? But if you want to compare with dengue vaccine, they both have different issues when we come to develop the vaccine for these two diseases. So we cannot really compare them with dengue vaccine. But in terms of COVID-19, we are to see what will come out later. But we need to answer a few questions, which how efficient is the vaccine, and then how long it will last, possibly. So if we need to get more shots after a while, that will be some answers that would take years or take some time to answer. These are the questions that would take years, mostly, to answer. So, yeah.
Yeah, thank you. I think the next question is also for Dr. Pang. So let's go on to the next one. Oh, I think this is already answered as well. The mutations affecting the creation of a vaccine. The next one by Mr. Leong, I think we leave to Dr. Pang, as well as the next one. Okay. Any others?
Yeah. So the rest is for Dr. Pang.
For Dr. Pang. Okay, but there's one from Dr. Nadia. I can't see it. Can you flash it on?
Nadia.
Dr. Nadia just posted a question in chat box. It says, thank you, Dr. Yap, for the clear explanation to the questions. It will also help us in trying to clear some speculations among, I can't see anymore, among
the public
and
family members.
Oh, it's just a comment. Okay, thank you. Thank you for the comment, Dr. Nadia. So now we go over to Dr. Pang.
Prof. Niao, can I ask Dr. Pang a question?
Yes, please do.
I just have one very practical question. It's related to the severe inflammatory response. We know that it will be associated with a lot of inflammatory markers, pro-inflammatory markers. Is there any possibility of looking at specific markers or group of markers to actually predict patients who are likely to suffer from this severe inflammatory response and do an early intervention? Just out of curiosity, I'm just wondering whether you can do this to help improve the outcome of patients.
Yes, thanks for asking that. I think, yeah, it's a great question. Obviously, in real life practice, we would like to actually predict which patient is going to deteriorate or going to actually do worse, and whether any early intervention will reduce the disease severity and to ensure the best outcome of the patient. I think I've shared with all of you just now regarding some cytokines and some inflammatory markers. I think just now, besides maybe we'll come back to this one again. One of the important markers is C-reactive protein. I think we are doing it right now as a kind of like routine for any patient who are admitted for pneumonia or just admitted for any COVID infection. I think it's a very good predictive marker. C-reactive protein is easy to do and cheap to perform. So apart from C-reactive protein, the other thing that we perform is the ferritin. In the interest of time, I didn't actually manage to share everything. Ferritin is another inflammatory markers are correlating with the severity of the disease. The cytokines I've shown you the interleukin 6, which I think has certain treatment implication. But there are also other interleukin that has been studied, like the interleukin 1, and there were some drugs actually being used for the interleukin 1 as well. And then the D-dimer test has also been shown to be associated with the severity of the disease. So these are the few things that we do, quite routinely in clinical practice to sort of like to gauge the severity of the disease. Obviously, if the patient have high inflammatory markers or high level of cytokines, even though the patient currently is not that severe, but we will have to ensure that the monitoring needs to be a lot more frequent and closer. And in fact, sometimes maybe certain treatment can be instituted early before the patient deteriorate up to the extent of irreversibility. Yeah.
Thank you very much. That's good to hear.
There's another raised hand. Dr. Jeffrey Niao.
Yes, Prof. Pang, would you comment on the use of remdesivir in this treatment regime?
Well, remdesivir, I think is such a controversial drug, right? I think the study done in China initially, of course, I think they did not manage to finish their intended recruitment, partly because the disease in China was, I think, under control much faster than what they can recruit. So the intention was to recruit, I think, more than 400 over patients who had severe disease, who require ICU care, who had ARDS. But I think they didn't manage to actually get the number. They only managed to get half of the number, and the disease is already under control in China. So based on the analysis of whatever patient they have recruited, there was no actually benefits seen among those treated with remdesivir compared to those who did not receive this drug, the so-called control group. But there was also some criticism regarding actually the study, partly because I think at the peak of the disease in China, I think a lot of patients had difficulty in getting the ICU bed, and hence many patients are waiting for the beds. So that could actually partly contributed to the poor outcome. And then, of course, I think you have another study done in US, the ACCT study, whereby it has been shown to add some actually benefit in terms of shortening the disease, the stay in ICU. I think 9 days compared to 11 days. So about 30% improvement. But they have shown that I think the drug also has been used for other studies and has been shown that patients with mild disease who did not require oxygen, although they have pneumonia, or they don't seem to actually benefited from remdesivir. And for patients who require mechanical ventilation, so say the actually disease is very severe, require mechanical ventilation, in that cohort of patients, they also don't improve compared to, I mean, the arm that is receiving remdesivir didn't actually improve compared to those who did not receive. So now I think the role of remdesivir, what I can see is only limited for patients who had severe pneumonia, who require oxygen therapy, but who are not severe enough to require mechanical ventilation. And in order to start, they have to start, I think, during that kind of like window period. And but the improvement is just very modest, I would say.
Yeah, thank you very much.
Yeah,
Welcome.
thank you. We now have a question, a raised hand from Dr. Wissam Abdul Kadir Yassin.
Yeah, good morning. I actually have two questions. First question is, does hypertensive patients on angiotensin converting enzyme inhibitors is protected from COVID-19? This is my first question. Second question, is there any scoring system which divide the patient infected with COVID-19 to mild case, moderate case, or severe case? It is a general practice that many diseases, we divide them into mild, moderate, severe cases according to a scoring system, depending on the clinical picture plus the laboratory investigations. So this scoring system, I believe will be very important to treat those patients who presented early, and we consider them according to, for example, laboratory investigation, high reactive protein, but still not manifested clinically well. We can divide them in, we can prospect that there will be severe cases to start intensive prophylactic treatment early. So to sum up my question, is there any scoring system which we can apply to divide COVID-19 patients to mild, moderate, severe?
Yes, I think let me just answer your second question first. Yeah, there are some actually classification on how we view the disease in terms of severity, but there's no universally accepted global classification. You look at WHO has certain way of classifying the disease. The US CDC has certain way of classifying. Even in Malaysia, we have certain way of classifying the disease. But I think by and large, I think one particular, and then in US, there's, and not just CDC, I think the Infectious Disease Society in US also have their own way of classifying. What I like is actually the Infectious Disease Society of America, the IDSA classification. They actually divide the patient into patient with mild disease, moderate, and then the severe. And then for patient, mild is, and then of course, I think that mild will also include the patient who did not have any symptom or had very mild symptoms. And typically, these are the group of patients who are on X-ray do not have any evidence of pneumonia. They are just having some symptoms. They may have some fever, and clinically they are very stable. Okay, so this is considered mild. And then patient with moderate disease, I think we can divide them into those who have evidence of pneumonia, but the one that is with milder form of pneumonia, they do not require oxygen therapy. Their SpO2 generally can be maintained above 94% under room air without any oxygen therapy. And the other form will be actually patients with pneumonia with a slightly more severe disease. They require oxygen, but hemodynamically they are stable. They do not require high flow oxygen. They do not require intensive care. They do not require other invasive or non-invasive therapy. And then after that, at the other spectrum of the disease, where the patient had more severe illness, where they require ICU admission, either because of the severity of the pneumonia or some other organ dysfunction. And then the vast majority of these individuals, I think they have a respiratory distress or pneumonia that is severe enough to be under ICU care. So those patients under ICU care will be those who require high flow oxygen. Then the much more severe cases will be those who require non-invasive, as well as those who require mechanical ventilation. So I think this is how we actually classify our patient. But I think severity is just one thing, but also we need to look at the speed of the deterioration. For instance, like if the patient is deteriorating very rapidly, then we foresee that this group of patients, they are going to, maybe from mild to actually become very severe very soon. So, so this group of patients, we may want to transfer them or maybe initiate certain therapy early. For instance, like if they come in, the X-ray was clear, and then after that the patient complained of some shortness of breath, and then you do the SpO2, they are actually dropping actually very quickly within a day, or then the X-ray started to see some pneumonic patches, then the PF ratio is also deteriorating. Then we know that this group of patients, they are quite likely to actually to deteriorate fast, as well as having a more severe disease in a very short period of time. Okay, so one is actually the current status. Two is actually the speed of the deterioration. The third thing that we consider is actually the time when they have this manifestation. So the question is, if I'm seeing someone who is still in the early phase of the disease, let's say end of the first week, and they are complaining of some shortness of breath, let's say on the day 6, shortness of breath. So then we have to watch very closely that that this patient has a very high likelihood of actually further deterioration, because we know that actually in general, most patients take about 2 weeks before the disease is under better control. And hence, if they had this deterioration at the end of first week, then we foresee that they are going to continue to deteriorate. On the other hand, you have a patient who are already 2 weeks or more than 2 weeks into the after the symptom onset, and now they're complaining a little bit of a shortness of breath, and we know that they are probably at the verge of progressive improvement. So this group of patients, we are not as worried as the other patient. So I think coming back is actually the current status, speed of deterioration, and then the time where actually the symptoms develop. And also we could actually do the imaging as well as the laboratory tests. I mentioned quite a few that we routinely perform in the clinical practice will be the C-reactive protein, the ferritin, and also for those with pneumonia, sometimes in certain hospitals, they can actually do the interleukin-6. So these are among the things that we actually monitor. Yeah. And of course, the EBG.
Yeah, thank you very much.
Welcome.
Thank you, Dr. Pang. Maybe we can go very quickly through the next few questions because we're really short of time. They're all concerned with treatment. So the first question from Dr. Wissam is, would you give ACE inhibitors to protect against COVID-19?
Actually, my question is, those patients, hypertensive patients on angiotensin-converting enzyme inhibitors, are they protected from the virus?
Okay, are they?
Okay, it's interesting. I think that this is actually a question many of us are concerned. I think there was some early concern that because of the nature of how the virus enter into the body, the ACE through the ACE2, right? And then the ACE inhibitor, generally they block the ACE1. So the question is whether by blocking all this, the receptor, whether it will make the individual a lot more likely to have a severe disease. And the question is whether we should stop the ACE inhibitor for patients who are already on the ACE inhibitor. But I think now we have good evidence to show that no, I think they are not at increased risk of disease severity. In fact, actually, when they did the analysis in actually a large cohort of patients, I think there was a published articles showing that in fact they are not at increased risk of dying. They are not at increased risk of complication. They are not at increased risk of cardiovascular complication. So now we have good evidence to show that for those who are already on ACE inhibitor, they should continue their ACE inhibitor because they are not at increased risk of any cardiovascular complications. So this is so much we know so far. Yeah, whether if the patient is not on the ACE inhibitor, and if you need to treat them for their hypertension, whether we should initiate an ACE inhibitor, I think that will be still debatable. But I think for patients who are already on ACE inhibitor, now we have good evidence to show that they do not need to stop it. They can continue. They are not at increased risk. Yeah.
Okay, so next question from Dr. Nagam.
Good afternoon. Okay, my question is patient who recover from the virus. Is there any possibility to stay a carrier for the disease and spread to the community?
Well, yeah, I think it's another great question. At the moment, we do not have any data to show that the virus has a chronic phase, whereby they just colonize the airway without causing any symptoms to the host, but they have the capability of transmitting to the other uninfected individuals. We do not have any solid evidence, but based on what we found so far, the published evidence, it seems that after 1 or 2 weeks, especially after the 2 weeks, I think most individuals are rendered non-infectious. So I think it's reassuring to know that patients who have recovered from the sickness, I think based on the current known data, the risk of transmitting the disease to another uninfected individual, if at all are there, are very, very low. Because most of the time, we can't isolate the virus in the culture after about a week after the onset. Although, I think in the reported literature, the PCR test may be positive up to about 6 weeks. But we know that PCR is just testing the genetic material. It's not talking about the live virus. So the virus, I think, could only be grown in the lab for up to about a week after the onset in the upper airway, up to about 10 to 14 days in the lower airway. So I think at this juncture, we can quite safely say that after 2 weeks, even if the PCR test is positive, most of these individuals are rendered non-infectious, and the risk of transmitting to another individual is very, very low.
Thank you, Dr. Pang. There's a related question as well I'm putting in now. What damage, is there any permanent damage after recovery?
Yes, I think just now I showed the autopsy of the lungs. It seems to show that in some individuals, there are significant damage to the lung tissue, the alveoli, as well as the blood vessel. So we would expect that the patients who had severe pneumonia, particularly those with ARDS, even after they have recovered from the infection, they would have residual lung damage. And this may take time to recover. Whether they will recover to their baseline is all dependent on how much damage has been made by the virus. For some patients, we expect them to recover to near normal. Some of them may have severe residual lung damage. So at least for the lung, I think we know that some of these are not reversible. So it is very important that during the when the patient is having the infection, adequate supportive care need to be instituted to minimize some of this lung damage. And then the next question is some of this damage are probably caused by the secondary bacterial infection. Some of them are caused by maybe improper mechanical ventilation. Some of them are caused by the thromboembolism that we actually talked about just now. So now I think for any patient who require admission and they have pneumonia, if there's no contraindication for anticoagulation, we will generally give them the so-called the DVT prophylaxis or VTE prophylaxis, the venous thromboembolism prophylaxis. And if they have severe disease, like if they have ARDS, in fact, I think there are some the expert society that suggests that perhaps we should give this group of patients a full anticoagulation therapy. The reason is because some of these patients, despite given the prophylactic therapy, they are still developing the thromboembolism. So suggesting that some of these patients, they may require the full dose of anticoagulation rather than just a prophylactic dose. Yeah.
Thank you, Dr. Pang. We'll just have two last questions, one from Dr. Jeffrey, who's been raising his hands regularly, and last one from Dr. Wissam. So, Dr. Jeffrey?
Oh, yes. For Dr. Pang, certain patients develop lingering fever after recovery.
Yes.
Would you comment on the possible causes of this?
Well, yeah, thank you for asking that. So the question is, why do some patients have lingering fever, or sometimes the fever seems to have resolved, and then after that the fever started again? So now the question would be, do they have a resurgence of the infections, or do they have other problem like a secondary bacterial infection, or do they have some other problems? Because fever sometimes can also arise from the tissue injury. I think we have to treat all these patients on a case-by-case basis. So I think we have also seen some of our patients who actually develop a new fever after they so-called declared recovered from the illness. But for many of these patients, I mean, luckily, even if they have some mild or low-grade fever, some of them do not actually seem to deteriorate further. And most of these patients do quite well with just a conservative observation. So I think we still do not know the whole picture. But in clinical practice, we always try to investigate whether there are some other causes apart from COVID-19. Yeah, but if it's due to COVID, if it's a PCR, it's just if the PCR test is positive, but it's at the low level, usually we will ignore that. And if the patient are quite stable, I think we can afford to just watch and see. Yeah, we don't really have to do anything.
Thank you. I think a question from Dr. Wissam first, and then we go to Dr. Nagam. Okay, Dr. Wissam, are you still there?
Yes. Thank you. My question is already answered. Thank you so much.
Okay, thank you. So, Dr. Nagam?
Okay, my question is ferritin. Why they use it in the hospital as a marker or one of the investigation for the COVID-19?
Yeah, ferritin, I think is not a specific marker. It is a marker for inflammation. It's like the acute phase reactants. So we see that for any patient with any infection, the ferritin generally will be raised. So this is used as a so-called predictor, and also to gauge, maybe to a certain extent, the underlying disease, how severe they are, or how likely they are going to turn severe. Although it's not a linear curve, but we can actually safely say that the higher the inflammatory marker, the more likely the patient is going to suffer from the more severe disease. So that's the reason why the ferritin is used. It's more like a monitoring marker. It doesn't have actually have a drug that we can actually target these molecules. It's unlike like interleukin 6, for instance, or interleukin 1 that you might want to develop specific molecules to suppress the cytokines. So this is slightly different. It's more like a monitoring biomarkers. Yeah.
Thank you. So I think we have to
Thank
come
you
to a
very much.
close. Yeah, but just one last question, because many people want to know this. Why do patients put on the ventilator die shortly afterwards? Is it because ventilation is not effective, or is it because the patients are too far gone to be saved by ventilation?
Yeah, thanks for the question. Yeah, I think it's, people tend to blame on actually the final puff or whatever we do at the end of the road sometimes. First, in the first place, I think we have to ask ourselves, why do the patient need ventilator? Obviously, the patient is having very severe disease that even with supplementary enriched oxygen therapy, we could not get enough gas exchange, and hence the patient need to be put on the mechanical ventilator, so like to support the patient ventilation, so that they have a chance of surviving the disease. So essentially, we are talking about a specific group of patients who have very severe disease. And if you look at the autopsy of this patient, you will see that they have very extensive epithelial damage, alveolar damage that I just highlighted in my slides, as well as the vascular damage. And also not to forget that when all this damage are occurring, okay, not only gas exchange will be affected, but they also have other implications. As we actually put in the patient on mechanical ventilation, they are also more likely to get the ventilator-associated pneumonia, meaning that they may get a secondary bacterial infection. And then the ventilator itself, to a certain extent, sometimes if it's not managed properly, can also cause some mechanical damage to the remaining lungs. So it's actually quite complex. Well, I think this is I think the patient, if they don't make it, is partly because of the severity of the disease. Although I think from our end, we're trying to minimize all the potential complication that is associated with this modality of therapy. Yeah.
Thank you, Dr. Pang. And thank you to everyone who stayed right to the end. Oh, Dr. Nagam, you want one last question, please. Yeah.
My question that since we have AC1 inhibitor in the market and it is very cheap medication and available, is it difficult to do AC2 inhibitor medication? Manufacture AC2 inhibitor medication?
Yeah, I think people have actually explored this, whether we could I actually give them, the patient, some inhibitor for ACE2 just to block the entrance of the disease. But the problem is when it is when you have an ACE2 inhibitor, the problem is we do not know, first of all, how effective it is. Secondly, it is only effective if it is administered on the so-called nebulized form. I think if the patient is taking it orally, it's not going to be effective because the interface is air and the epithelial. So the drug needs to actually coat the epithelial to prevent the virus from actually coming in. But if you are using it the nebulized form, then we are talking about administering this prophylactic therapy to the entire population on a daily basis. So I can't think of actually the skill that we need to employ. So while I think in principle it seems a very interesting model, and it may work if we find actually the effective inhibitors. I think it's not probably difficult to actually manufacture the inhibitor. But the problem is you need to administer them into the lung through the nebulization. And we know that if you administer any drugs through the lung, it could by itself create a problem by itself, meaning that sometimes they can cause bronchospasm. And so, yeah. So although in principle it seems like it's an interesting model, but in real life, I think it's still quite difficult to implement. Yeah.
Thank you, Dr. Pang. Thank you. Yeah, I'm really reluctant to stop this session, but I think I have to release our speakers. They've had a long day. If you have any more questions, I think Dr. Pang and Dr. Yap would be happy to reply your questions sent to them in writing. So it's time now to close the session. Thank you everybody for attending and for staying to the end. Have a nice weekend.
Thank you very much. Yeah, a pleasure to be here.
Okay, thank you.